Hi guys, in this video I will be explaining how you guys can synthesize insulin in your home lab. This idea came from my auntie who said that instead of synthesizing neurotoxins, I should make insulin, which is more needed. Additionally, if this video gets 10,000 likes, I will try synthesizing insulin at home myself. Hit that subscribe button and yeah, let's get into it. So the general steps for insulin production includes gene cloning and transformation, where we have to clone the insulin gene into a suitable plasmid vector and then transform the plasmid into a competent host cell such as E. coli or yeast. And from there, we will grow the transformed cells in a nutrient-rich solution, either agar or GH broth. And from this, we will induce penicillin production using the appropriate inducer. From this, we will then harvest the cells and then lyse the cells to release the insulin, and then we'll use some other techniques such as centrifugation to produce the insulin, and then some quality control. That's about it. And below here is a link of NIH, and it details how they made insulin from bacteria. But yeah, that's about it. So first, we have to obtain the human insulin gene sequence from a genetic database or through DNA synthesis. This will be delved into. This is another step where we have to contact a gene synthesis company, such as Integrated DNA Technologies or Thermo Fisher Scientific, and provide them with a sequence. They will then be able to synthesize the gene and deliver it to us. From here, we will do some PCR amplification from cDNA and isolate the mRNA. This is from the DNA. And so from this, we will use kits like Qiagen RNeasy kit to isolate mRNA from human pancreatic tissue or a cell line that expresses insulin. From this, we will then do a reverse transcription using Thermo Fisher's superscript IV to synthesize the DNA from the isolated mRNA. From this, we will also do PCR amplification and use primers specific to the insulin gene, which can be found online. And right here is the forward primer and the reverse primer. From this, we will perform PCR using these primers and a high fidelity DNA polymerase, which can be purchased from online. The human gene is also located on the short arm of chromosome 11 at position 15.5. So I don't know what the fuck you want to do, but maybe you can just go take a little blood sample and just fucking somehow conjure that shit up. Do what you want to fucking do, bro. I don't give a shit. Or you can just buy this online for $61, bro. This shit is a steal. From here, we will then use the correct vector. The vector should contain regulatory elements for expression in yeast, including a promoter, terminator, and selectable marker. We will then clone the IGF-I gene into the expression vector by inserting the amplified human IGF-I gene into the yeast expression vector using standard molecular biology techniques like restriction enzyme, digestion, and ligation. So from this, we will inoculate the transformed yeast cells into a liquid growth medium containing appropriate selection markers and grow them to mid-log phase. From here, we'll add an inducer such as galactose for Cerevisiae or methanol for P. pastoria to the culture to initiate expression of the IGF-I gene. Then we incubate the induced culture for a suitable period to allow for the protein expression. From here, we will then cultivate the positive yeast colonies in liquid medium supplemented with appropriate nutrients and inducers, and we will produce one that is suitable for the yeast. And from this, we will then add more inducers to a suitable concentration to trigger the expression of the IGF-I gene and promote for more insulin production. Now we have to incubate the culture under conditions that are optimal for this protein expression, including temperature, pH, and aeration. And with the temperature of the culture being around 30 degrees Celsius, and with a pH between 5 to 6, and moderate to vigorous aeration by shaking the culture at around 200 to 250 RPM to ensure there is continuous agitation to promote oxygen transfer and prevent oxygen limitation.